Hello and welcome to the second webinar in the Organic Seed Production 6-webinar series organized by the Organic Seed Alliance and the Multinational Exchange for Sustainable Agriculture. We'll be hosting one webinar in this series on the third Tuesday of each month through November 2016, and you are welcome to attend as many of them as you'd like. This is your host, Alice Formiga from the eOrganic Community of Practice. Today's presentation will last about 45 minutes, and then we'll have 30 minutes for questions. Today's speakers are Jared Zystro and Lori McKenzie of the Organic Seed Alliance, and their topic is Trials and Selection. Jared and Lori will discuss how they conduct on-farm variety trials and how they do field selection or roguing to improve the performance of open-pollinated seed crops. So without any further ado, I'm going to hand the microphone over to Lori McKenzie.
All right. Good morning, everybody. Welcome and thank you for joining us. So I'm going to spend about 20 minutes with you guys talking about trials and what trials do for you and how you set them up, and then I'm going to hand it off to Jared and he will talk to you about roguing and selection and some more aspects of doing trials. So the first question is, why do you want to do trials? Trials are really helpful for a number of things on your farm. Probably the most common reason to do trials is to identify the varieties that work best for you. So this can be, again, for a number of things. It can be disease resistance, it can be resistance or tolerance to some abiotic or biotic stress or pest. It can be to fill a market niche. It can look for varieties that extend your season, either in the early spring or the fall. Varieties that yield better than others, varieties that are unique, and it's also a great tool to use for identifying breeding material, which plays into seed production. If you are looking to do any kind of breeding work or just to do profitable seed crop production, trials can help you figure out which crops will produce a viable, mature, high-yielding crop for you. So when you're planning your trial and thinking about what your trial is going to be for and what it's going to do for you and what kind of information you're going to get, here are some of the things you want to think about. You want to prioritize your crops. Which crops are you going to do trials on if you're going to do more than one? And what are the goals of your trial? So these probably seem very obvious, but it's easy once you get a trial in the ground. You'll start seeing all sorts of things and all sorts of differences that crop up that you were maybe not anticipating. And identifying your goals when you start out can help you focus and figure out what you really need to pay attention to in your trial. You can take lots of data in a trial and not all that data will be useful to you. So really sticking by what your goals are can help you streamline your work and be more efficient with your trials. That said, of course, you always want to keep your eyes open for the unexpected and the interesting. Another thing to think about when you're planning out the trial at the beginning of the season is what, when, and how will you evaluate, and what kind of tools will you need to do those evaluations. I will come back to this towards the end of my talk, and we'll talk a little bit more about how to evaluate and means for evaluating, but you want to think about at the outset, are you going to be taking notes and just annotated notes on things? Are you going to take measurements? And if so, are you going to need tools to take those measurements? Are you going to take yield measurements? Are you going to take plant height and width kind of data? So that plays into your goals and what's important to you. So you want to think about what you're going to look for, how you're going to do it, and set yourself up to succeed from the beginning. Think about what kind of data sheet you're going to want, if you're going to want a data sheet, or if you just want to have a notebook and take notes, because I guarantee the trial will sort of all of a sudden you will have something to evaluate, and it's better to be prepared ahead of time. It will save you a lot of stress. When you're thinking about what to include in your trial, you always want to include what the popular and important commercial varieties are in your region or in your market. So if you're a wholesale producer, you may be looking at the varieties that are most commonly grown for the wholesale market. You may be looking at the varieties in your region that grow really well. It's good to include both open pollinated and F1 hybrid standards. If you can get them, that's more easily done for some crops than others. If you're interested in doing any breeding, it's good to include any potential breeding material or any projects that you're working on. That gives you the opportunity to look at that material against the commercial standards. And if you're looking for any sort of disease or abiotic or biotic or pest resistance or disease tolerance, it's great to include a variety at least or several varieties that you know are both susceptible and resistant. Disease is a little challenging to capture in a trial, but if you can look at one plot that is obviously susceptible and is expressing a lot of the disease or whatever it is, the stress that you're looking at, right next to a plot that is not expressing it and is very resistant or tolerant of it, that will give you a great visual of the rest of to compare the rest of the material in your trial. Another thing to think about is, in terms of your goals, is what do you want to know and how much are you willing to do? If you're doing experimentation, that means you're doing replicated trials, and that's the only way to tell if you have statistical differences between your varieties. And what a statistic tells you is whether the difference, let's say a difference in yield, is due to chance or due to the fact that the varieties are actually different from each other. So doing a replicated experimental trial is the only way to figure that out. You don't have to do that. You can do what's called an observational trial, and that's where you have one rep, possibly two, and that will give you good information about what the varieties are doing or how well they're performing for you. You just won't be able to tell if they're really different from each other or whether there are minor differences in your soil or your irrigation or the fertilization, or maybe one plot happened to get a slightly larger amount of compost than another plot. Doing the replicated trial not only lets you determine the statistical differences, it also will bring out those differences, and we'll come back to that in a minute, which you can't get with an observational trial. Now, if you do a replicated trial with experimentation, you are going to need to take data and you are going to need to do data analysis, which involves using some sort of statistical program. You can do basic statistics in Excel. You can also use programs that are designed to do statistical analysis, such as R or SAS, but that's a whole nother additional element of work at the end of the season to find out what your trial has really shown you. So that's something to consider when you're setting up your trial. When you're doing an experimental trial and you're doing replication, you're also going to want to randomize, and we'll come back to that as well, but that means that each replication you're randomly assigning all the varieties to each plot, and I'll have a visual of that here in just a minute. So what constitutes an effective trial? We've covered some of these already, knowing your intention and setting your goals. You want to locate your trial in an area of your farm where you have consistent field conditions. So this, and we'll see a visual of that in just a minute, but that means thinking about a lot of different environmental factors. It means thinking about your soil, your fertility, your water, whether there's shade, any kind of differences in your field that might contribute to difference in performance you want to take into consideration. Replicating and randomizing we just covered. You want to use a block design if you're going to do a randomized and replicated trial, and I'll show you that in just a second. You want to use border rows if possible. This is not always possible because depending on the size of your trial you might not have space to add in border rows, but border rows give you the confidence of equal competition, meaning that all of the plots in your trial have that same crop around them on all four sides. So instead of having a row, say you're doing a trial of carrots, instead of having your carrot trial right next to, let's say, a row of cabbage, you have that carrot plot and then you have rows of carrots on each side. So all sides of your carrot trial are having equal competition with other carrots, instead of a different kind of crop. It's very important to label. Go ahead, Jared.
Sorry, just to butt in. The other advantage of border rows is that the edges of your trial are often the places that don't get treated quite the same. So it might be that they don't get tilled quite as thoroughly, or the overhead irrigation doesn't quite reach, or the manure spreader made a turnaround and didn't quite make those corners. So the border rows often are places that may not quite get the same treatment as the middle of the field, and so having the borders helps protect your trial in the middle.
Great. Thanks, Jared. I've invited Jared to jump on any of this to add in his thoughts and expertise. And when you evaluate the trial, whether you're doing a replicated experimental trial or an observational trial, it's important and recommended to evaluate the trial blind, which means your evaluation sheet and your markers in the field do not tell you which varieties you're looking at. And even if you think you don't have any feeling one way about the varieties in your trial, or whether you think you might like one more than the other, it's very, very hard to maintain what the word I'm looking for, to not have a bias against one or the other. And even if it's unconscious, you may like the name of something, or you may like the person who bred it or the company you got it from, and that might influence your evaluations on something if you know the variety you're looking at. And I'll show you some ways to do that, and I'll show you some data sheets that I use. We'll talk a little more in depth about scoring and measuring and recording those important traits, and then the data analysis to get the information of what you learned from doing your trial. So when you're doing an experimental trial, you're doing randomization and replication. 3 replications is very standard for this kind of an arrangement. You can do more if you want. Less is not recommended. If you lose a rep, then you only have 1 or 2 left, and then you're doing an observational trial. Your first rep can be in order, and that's in quotation marks, because you can determine whatever that order is, whether it's alphabetical or you have any kind of order. The first one, it actually doesn't matter. What you think of as being in an order can essentially be considered random for your first rep, but then you want to mix up your other replications. And here is an example of a block design. So this shows you both the randomization and the block design and the border. So in the one that you're looking at, when you're looking at your screen, the one on the left, actually I think I can use my pointer here. So this is your rep 1, yellow is your rep 1, red is your rep 2, and blue is your rep 3. So this is what I'm talking about with rep 1 being in some sort of order, and then you're randomizing your second and third reps. And the way that I randomize when I put a trial together, I do it in Excel, and I put 1, 2, 3, 4, 5, and 6 in a row, and then I have a column right next to that, and I use a random number generator, and then I sort my 1, 2, 3, 4, 5, 6 based on the random number column. And then I just put that straight into my trial design as the next rep, and then I do it again, and I put that one in the next slot. Now, I want to point out that you'll notice these are oriented in 2 different ways. And if you do have variation in the field that you are aware of, whether it's shading or soil differences, it's best to try and capture that difference by rep, and that's what the block design is. So if I have, let's say I have a border of trees over here on the edge of my field, and my rep 1 is getting more shade than rep 2 and 3, I would want to choose this orientation, because then if there is any effect of that shade, that should show up in your analysis as a block effect, and you can manage that in your statistical analysis. Now, this is another way to do it if you have any known, let's say you have a field soil gradient. running from, let's say this is north to south, then you'd want to capture as much of that variation in each rep as possible. So you would orient this way in horizontal blocks instead of vertical blocks. So here's an example of what that soil variation can look like. If you were going to design a trial to fit in this field, you would look for the most consistent area to locate your trial in, which would probably be about here. And notice this, we've got those longitudinal blocks instead of vertical blocks, so we're capturing this amount of variation by orienting our blocks the long way along the field, and then we're putting in that border row all around the trial. When you're thinking about where to place the trial in the field, we've talked about soil type and irrigation, cultivation as well, fertility, harvesting. It's always ideal to locate your trial in the middle of a production of the same crop if possible. So if this is obviously a carrot field, if you could locate your trial here in the middle of the field, then you've got border rows that are already a part of your production system. Another great benefit of doing that is then your trial is sure to be treated as the rest of your production crop is treated. You're not treating it or putting it under conditions any different than your typical production. If you are doing that experimental trial where you have replications, you always want to strive to do everything to all of the reps. Let's say you have 3 reps. You always want to do anything to all 3 reps on the same day if possible. If that's not possible, you want to do whatever you're going to do to 1 block at a time. Let's say you go through and you're weeding and you can only weed rep 1 because it's the end of the day. Then you want to just weed rep 1, and let's say you don't get back to it for another 3 days, then you come back and you do rep 2 and rep 3. Instead of doing as much as you can and getting rep 1 and maybe half of rep 2 done, and then coming back and finishing that rep 2 and rep 3 a few days later. Now, it probably sounds like that's not going to be much of an effect or terribly important, but you don't know if it will be or if it won't be. And if you treat by block, then you can go back and you can figure out if you have any differences that show up in those as block effects. You can kind of go back and figure out what that is if it's not a variety effect. Some other things to consider, if you want to, you can orient and locate your trial in not optimal conditions. This is especially if you're looking for varieties that will perform well under stress conditions or disease conditions or drought conditions. So there's kind of 2 options here. One is to do that, to locate your trial in a more challenging location, and that those non-optimal conditions will reveal your workhorse varieties. They'll reveal the varieties that can really handle and tolerate those challenging conditions. The other option is to grow the trial under the conditions of intended use. Whatever your normal production standards and cultivation and practices are, and then you'll find the varieties that really thrive and rise to the top under your standard conditions. It's very, very, very important to mark and map your trial. You always want to make a field map of your trial. Don't just rely on markers in the field because they get lost, they get broken, they get run over by tractors, they get moved by cultivation equipment. So having a field map along with whatever kind of markers and indicators you have in the field is very important. One thing that can be very helpful, especially if you're doing a crop that's difficult to tell apart. If you're doing carrots and they're all orange carrots, and it would be difficult to tell where one plot ends and the next plot begins, you can put in breaks between each of your varieties, between each of your plots. That can be a physical break where you just leave a foot or 2 feet of an alleyway. Maybe you plant some pollinator plants in there. This is a fairly large-scale trial, so depending on how big your trial is, there are easier and less easy ways to do that, but we put in pollinator blocks sometimes. I'll put in phacelia or some sort of flower in between each block. You can also put in a different color of whatever crop you're doing. So if you have a carrot trial, you can put in a little bit of purple carrot between your orange carrots, or if you have multiple colors in your trial, you can consider setting up the trial so that each you have a different color from one plot to another. That will help you figure that out. Consider using multiple marking techniques, and these stakes are fairly short in this picture. If you have a crop that's going to get fairly tall, consider coming back and putting tall flags in, or putting tall flags along with your marker crops. Think about what the crop is going to look like when it's full grown and not just when you planted it. And it's also very helpful to orient your data sheet to your field map. And actually, I'll come back to that in just a few slides. This is an example of that multiple marking technique. So the picture on the left is these are both broccoli trials, and that's what the broccoli trial looked like when we transplanted it, and then the picture on the right is what the broccoli trial looks like when we're coming back to evaluate it. And it's very difficult to find those little orange tags, so it's helpful to have, you can kind of see behind the orange flag there's actually some taller flags which help identify where the plots end. But if those get pulled out, which they often do by tractors, if you're doing any mechanical cultivation, that's where the breaks can be very helpful. If you lose your tags and or your flags and you have a break, especially if it's an obvious break or you've planted flowers in between, it's still very easy to tell where your plots are. This is a field map that I use. This is one of my maps from this year, a tomato trial that I've done, and there are a few things I want to point out to you. So each of these it's the same thing. This picture over here on the left with all the numbers, that's my blind map. So when I'm going out into the field to do my evaluations, I only print that out. This one on the right are all the varieties, and this is what I use when I plant the trial. So it's easy for me to tell where everything goes. And I like to color code things. It's really great if you have a color printer. It just makes another visual cue for you of what rep you're in. So you can tell in this one, my first rep is green, my second rep is blue, my third rep is pink. And then when I set up my data sheet, I start here, and these are, all of my rows are trellised and they're on black plastic. This is a trial that I do not have border rows on because I did not have enough room in my field for more plastic, and I didn't have enough trellising set up to do entire border rows around the trial. However, I do have border plants, just a few on each end, just not on the edges. So because this entire line is almost 400 feet long, and I've got T-posts in, especially when the plants get big, it's not easy to walk across the row. So I start here and I do my data evaluations, and I walk down the row taking all my notes and my measurements, and then I turn around and I come right back, and then I can do these plots. And then I come out at the end of my trial, and this is a serpentine setup. It's very amenable to doing your data evaluation and very helpful to set up your data sheet in a way such that you can walk through the field and easily take any data. It will save you a lot of time, and it will save your brain a fair amount of confusion. So when you go out to do those evaluations out in the field, you are thinking about what's important to evaluate, when are you going to go out and take your evaluations, and what do you need to bring with you to collect that data. I recommend scoring your trial multiple times, and of course this depends on your objectives and what your goals are for the trial, but you can start very early. You can take your first set of evaluations in the flat if you're doing transplants, or out in the field during emergence. You can take early vigor evaluations. Again, I highly recommend don't take a lot of data that you don't need. Just kind of be swimming in it, and it doesn't actually help you meet your trial objectives. You want to think about, are you measuring things or are you scoring things? And we'll come back to this on the next slide. I'll go a little bit more into depth on that. And project the dates that you're going to be needing to go out and score and evaluate your trial. It's easy to let the trial get away from you, especially when you have lots of other things going on your farm. So I put evaluation times on my calendar, on my digital calendar on my computer. I write them on my calendar at home, so I don't have to keep it in my mind all the time, and I can have an easy access visualization of, oh yeah, I need to go out and look at corn silking about now. These are some typical things that we tend to evaluate in our trials and are common among most trial evaluations. So looking at, how long it takes to get to maturity, what's the yield, color, flavor, texture, storage life, uniformity, and uniformity can be on a plot basis or a plant basis depending on what kind of crop we're looking at. So how to assign a score for each trait. So these are traits that you would not be measuring. So if you were measuring like plant height, you would go out with the measuring stick and you would measure it's 17 inches tall. And these are traits that you're not going to measure quantitatively like that. This is disease resistance or pest resistance, or you can do color this way. Generally this can be used either in a relative or an absolute way, and relative meaning you're comparing each of the varieties within the trial to all the other varieties within the trial. Absolute meaning you have a scale that you've set, you go out into the field with it, and I'll show you an example of that next. So we generally use a 1 to 5 or a 1 to 9 scale. We have 1 being the poorest of whatever you're evaluating and 5 or 9 being the best. We just discussed that. So if you're doing a relative evaluation, you want to walk your entire trial and find your 1 and find your 9 or your 5, so your worst and your best plot. Walk through your entire trial, find those plots, and then that sets the scale for you. And then when you go back, you want to use all the numbers in the range if possible, because then you'll be able to see differences across the trial. Here's an example of a scoring guide that I've put together for one of my trials, for my cabbage trial this year. This is an example of an absolute rather than a relative scoring. So you can see here is insect damage. Here's my 1 to 9 scale. And then you use your even numbers essentially as tiebreakers. And I like to do this because I have found that sometimes it's difficult when I go out into a trial and I don't actually have a huge spread, and I don't want to force my plots to be different if they're not actually that different. So if all of my plots are 7 or a 9, then I want to capture that information. So this just depends on your preference and what the goals of your trial are. Here's another example. Photo scales are great. This is thrips damage on onions, and gives you a great visual of that 1 to 5 scale in terms of thrips damage. Here's an example of measuring traits with corn, measuring ear width and probably ear length as well. Record keeping, it's a great idea to keep lots of notes. Always start out with some sort of record log of the varieties in your trial and what your seed source was, keeping track of all of your important dates such as transplanting, seeding, any annotated notes. I find I'm most comfortable having both paper and electronic copies of things. Paper things are most easy to deal with in the field. They're also fairly easy to lose once you come back and they join all the other papers in your office. So I found it's ideal if you can get that information from the paper into your computer, and then you've got multiple copies, and you're less likely to lose the information. When you get to the point of doing your data analysis, if you're doing an observational trial, you're probably doing things like descriptive statistics. You're either looking at the mean, the median, and the mode. Again, here you have no statistical significance, but that does not mean you're not getting useful information. If you're doing a replicated trial, you're probably doing an analysis of variance. This is a straightforward, fairly easy means of analyzing data from replicated trials. It does require some sort of statistical software. I believe you can do it in Excel. If you're not a statistics person, you may need someone to help you, or you may need to do some research online and figure out coding. This is the only way to figure out if the differences you see are not due to chance, but are in fact due to a difference between varieties. Here is an example of some of that information. And the great thing about reps that I want to point out here is it also shows you which varieties are most consistent. So in this case, this is a carrot trial, and you can see Nelson. I'm not sure, this is carrot top height. So Nelson was a little more variable. Bolero was fairly variable. Scarlet Knots in this case is very consistent. So it's another nice thing about replicated trials is they give you some consistency data of performance. This is a kale trial that OSA did several years ago down in Southern Oregon, and this is just an example of how you can share data in a very easy way to look at. The darker green a trait is rated, the more better it is, and the lighter colored it is, the scale goes from green to red. So anything that's tending towards sort of this one down here is reddish orange, and then the overall is an average of the scores of all these different traits. So this is another way to present data, and it's also the colors make it a nice visual cue to show you which ones are performing best. And then to wrap up, trials are ongoing. You don't want to wait until the trial is done or nearly done to compare. In most cases, you will have missed out on some valuable information. You always want to be sure that you're comparing the best varieties available, and you always want to test the newest varieties from good sources. You can do source trials, and you can do the same variety of a crop from multiple sources. I guarantee you will see differences, even though it's the same variety. That can be a fun and interesting trial to do. It's nice to do 2 years of a trial if possible. You never know in 1 year if you had a poor performer, if it just had a bad year, or if it's actually a poor variety for you. So if you have the patience, the time, and the resources, it's great to include things in a trial for at least 2 years before you give them the boot. And you'll see the best results from your trials if you can do multiple trials across both years and locations. If you have enough area on your farm, you can move a trial around to a couple different places, and you can do it for a couple of years. You'll get the most robust information. And OSA has lots of helpful resources for you as you're endeavoring to do your own on-farm trials. These are just examples of some of the many publications we have. The On-Farm Variety Trial Guide will be a great resource for you going forward. It has pretty much all the information that we're covering today. And then the lettuce one on the left is just an example. We have many crop-specific guides on seed production and climate issues in seed production, and there's a lot of information there to help you. And with that, I will turn it back over to Jared.
All right, thank you, Larry. That was very helpful, and definitely something, trials are something that help all farmers, but definitely as seed growers, you're going to find a lot of value from being able to conduct trials, both for your own use to say, okay, well, which one of these varieties might be the most, one that I can most profitably grow for seed, and also to give really good information to whoever's buying your seed crops. So it's definitely going to be part of most seed growers' toolbox to be able to successfully conduct trials. So thanks, Larry, for doing such a nice job of presenting that. I'm going to be covering the next part of our talk today, which is going to be talking about selection. So I'm specifically talking about selection in the context of seed production. So if you're a seed grower and you are for most seed growers, you have a certain level of responsibility for maintaining the varieties that you're producing seed of and improving the varieties as well. And you can do that through selection. So I'm going to be giving a little bit of an overview of why select and how to select and some of the specific methods that you can use to do selection in your seed production. Basic definition to think of when we're talking about selection. Selection is essentially deciding which plants in the population of plants that are growing in your seed production field, which of those plants are going to be contributing to the next generation. Which plants are you going to be collecting seed from? Which plants will be contributing pollen to make seeds for the next generation? And by making good choices, that's how you're going to be able to either clean up a variety that may have some defects, maintain a variety, or improve that variety. So what are some of the reasons that you might be selecting? What are some of the things that you'll be selecting on? You'll be looking in any field. There's typically going to be some plants that are superior and some plants that are going to be inferior. Plants may be smaller. You may see some that are more disease-prone or may have poorer yield. And you may have some plants that don't match the varietal type. It's not necessarily that they're have a defect, other than they're just not the type that you're advertising as this variety. So they may have the wrong fruit color or seed color. They may be the wrong shape. They may have the wrong habit, like this picture here where you can see a vining zucchini plant in a field of what should be all bush type zucchinis. So this is something that you wouldn't want. Something to keep in mind when you're thinking about selection and when you're thinking about trueness to type and uniformity is that I think for many of us there is a real appeal to strive for perfectly uniform varieties. They look really pretty when the field is all perfectly even and everything is the same. Every plant is the same size and shape. It can be more convenient as well. But keep in mind that genetic diversity is something that as a seed grower you have a certain degree of responsibility to steward, because that the genetic diversity that's in your seed crop is what can potentially allow your seed crop to adapt to changing disease or climatic pressures in the future and reduce the risks that a given pest or disease might wipe out your whole crop. So think carefully about where you require uniformity in your seed crop and how much uniformity you require as you do selection, and use that to determine how severely you select the plant. Selection, first, really, you can think about it on the preventative side. So before we even think about how do we actively select the best plants in the field and have them make seed, there's ways that we can think about how do we prevent issues of contamination or off types or inferior types getting into our seed varieties in the first place. So first thing to think about is in terms of isolation distances, like we talked about last month when we were talking about production planning. Know what the isolation distance requirements are for your crop, so that way undesired pollen doesn't get into your seed crop. And keep those isolation distances in mind, both for your own crops. So, for example, if you have some spinach seed that you're growing and you've got a field of baby leaf spinach that you're also just harvesting for market, be aware, are they separated by enough isolation distance? If not, are you ensuring that that field of baby leaf spinach has been harvested and tilled in before it has a chance to flower and spread its pollen to your seed production? So think about it for your crops. Think about it for what crops are your neighbors growing, and can they potentially contaminate your field with their pollen? Keep in mind for any volunteers. So if you grew one variety of Brassica oleracea kale last year and you're growing a new variety this year, well, that's not a perfect example because the biennial. Let's say we can go back to the spinach example. If you grew some spinach for seed last year and you're growing a different variety of spinach for seed this year, if some of that seed from last year shattered and fell on the ground during harvest, are you making sure that there aren't little tiny volunteer spinach plants that are going to be shedding pollen and potentially contaminating your variety that you're producing this year? And finally, there's certain weeds that can cross-pollinate with your seed crops if they're the same species. So things like queen anne's lace and carrots being a classic example, but there's a number of other weeds that you should be aware of that can potentially be changing the genetics of your variety and making it less desirable. So that's one source of contamination that you want to be aware of and you want to select against. The other is in the physical mixing of seed. So being careful about cleaning all of your equipment when you're working with seed crops. So this can be everything from your combines to the little flaps in your tarps to the cracks in your seed cleaning screens. All of these places are places where seeds can get mixed between lots. And so one of the easiest ways to do selection is be able to improve the quality and uniformity of your variety and reduce the amounts of off types in your variety up front by preventative measures like these. The second topic that I wanted to talk about when thinking about managing selection is this idea of stock seed. And this is a really kind of a opportunity to take your seed production to the next level. And this is what most large commercial seed producers will do this to some extent or not. And when we talk about stock seed, essentially what we're talking about is this is seed that you're going to be growing specifically to be used as your own stock of seed to grow future seed crops. And this is opposed to production seed, which is the seed you're going to be growing and then selling to other farmers that will just be using it to grow produce or grains with it. And so stock seed is where you're going to be, if you separate out this in your mind and as you plan out your production, you have an opportunity to much more intensively select and invest more time in your stock seed because you can grow a relatively small amount of stock seed and that will supply your own seed needs potentially for multiple years. And so you can invest a lot in a small field of stock seed to make sure that it's the most superior plants are the ones that you saved seed from to make your stock seed, and then you can use that as your own seed for a series of years. You can do this two ways. You can either select plants to serve as your source of stock seed from within your production field. So this would be, for example, if you had a larger field that you were growing for production, you might go through and flag some real standout plants, some that show superior traits, and and save seed from those plants separately from the rest of the plants in the field. So come by and harvest those a little bit earlier and take those to the side and have those be your stock seed before you go and harvest the rest of the seed for sale. So have stock seed as a separate grow out. So you could be producing stock seed either in an isolated location or in a separate year. Then you grow your production seed, so that the advantage with that, especially when we're talking about cross-pollinated crops, is that if you have an entire field that you are just managing for stock seed, then you have the opportunity to select both the seed parents and the pollen donating parents in that field. Whereas if you have a cross-pollinated crop and you flag individual plants within a larger field to be your to serve as your source of stock seed, they'll still have pollen coming in from the rest of the field. So which way you manage your stock seed will depend on your resources. It will depend on the crop life cycle, whether it's cross-pollinating or self-pollinating. But either which way, just keeping in mind this idea of stock seed, of thinking about, okay, what is the seed that I'm producing for myself? And once you think about that, conceptualize that as a different thing than the seed that you're growing from everyone else. That will be a leg up that you'll have to start doing a better job of selecting. So question is when to select. And selection can and generally should happen multiple times throughout the season, because just as Lori pointed out when she was talking about trial evaluation, different phases of a crop's life cycle are going to present different opportunities to see clear differences between plants and make selections. So this can be all the way from the seedling stage, if you're looking at trying to identify plants that are able to germinate and emerge and grow earlier within their first few leaves, to be able to really select for that early vigor that will allow you to hopefully come in and be able to cultivate a little sooner and have plants that get a jump on the weeds. So that would be something you'd see really early in the season that might kind of get lost and be harder to differentiate as all the plants get larger later in the season. If you're looking for something like maturity of the plants, if you're either selecting for earlier or later maturity, often flowering time can be a really good chance where you can really clearly see plants that are the first to flower or the last to flower, and you might not really see that as well once everything's flowered. It'll be harder to gauge maturity at that point. There are opportunities for, there may be diseases that come through early in the season or later in the season that the plant, there may be times when it's most visible, the effects of the diseases on the plants. And so you want to kind of be walking through the field multiple times, all the way up till harvest and actually selecting based on the yield of the plant or on the size of the seed, or on, if you're talking about wet seeded crops on the fruits, or if you're talking about biennial plants, you could be selecting on the roots of the plants. So carrots, for example, when you're replanting them to produce, to transition from first year root production to second year seed production. So just thinking through, going back to planning, think through what some of those key stages are that will give you an opportunity to really see clear differences and make sure and get out to the field and make selections at those times. If you're selecting plants by removing inferior plants, then the best chance to do that is going to be to remove as many plants as you can before fall, because any plants that you don't want in your to be contributing genes to the next generation if you can remove them before they can spread their pollen, that's going to be more effective than if you wait until after they've pollinated. It's still better than nothing to remove them after pollination, because then they won't be contributing any seeds. But as many opportunities for selection before pollination that you can do, the better. So I'm going to talk a little bit about a few different kinds of selection that you might be doing and how you would do those. I'm going to be talking about negative mass selection or roguing. I'm going to be talking about positive mass selection, and I'm going to be talking about progeny or family selection. So negative selection, negative mass selection, essentially just is the act of walking through your field and removing typically a relatively low percentage of the plants in your field. So maybe 10, 15, 20% of the plants in your field that are inferior or off-type for some reason or another, you'd be removing those from the field. This is something that can be done in a production field, in a field that you're growing specifically, to produce seed for sale without having a huge adverse impact on your yields. This kind of selection is going to be most effective in self-pollinated crops instead of cross-pollinated crops, because typically what you're looking at if you've got, say, some off-type plants in a self-pollinated crop, if you remove them, if they were just off-types from something like seed contamination from, say, some equipment having some seeds in it, well, if you've removed it during this roguing or negative mass selection process, then those probably haven't had a chance because they're self-pollinated to be able to really spread into the population. Those off-type genes haven't really spread into your variety, and so you'll be a little more effective. Whereas in a cross-pollinated crop, especially if you weren't able to remove those off-types until after they've pollinated, then it's going to take multiple years to sort of remove off-types. Positive mass selection is the flip side, where in negative mass selection, you're typically looking at the plants that you don't want in the field. So a relatively small percentage of plants that you don't want for negative mass selection. With positive mass selection, you're looking at a relatively small percentage of the plants that you do want. So these are really shifting your focus to the best plants in the field. So this is going to be something that you're more likely going to be doing when you are able to invest more time and are less worried about the final yields of your production. So this would be, again, if you were doing stock seed production, you would be typically employing some sort of positive mass selection. So you might be picking the best 10, 15, 20% of the plants in the field. So some of the tricks with this, again, you're just identifying individual plants, and the challenge with identifying individual plants is that there are aspects of those plants that are due to genetics. There are reasons that those plants look better because of their genetics, but there's also a lot of aspects of why one plant might look better than another that are just environmental, due to maybe it had a little wider spacing around it, or it had a little more fertility or better water. And so you always have that caveat that when you're picking individual plants, part of what you're seeing is potentially because those plants are genetically superior, but a part of it might also just be that they had slightly better environmental conditions in the field that made them better. So some of the things that you can do to improve your selection when you're doing mass selection, as I've mentioned before, as much selection as you can do before pollination. The larger the population that you start with, the better. Again, with a very important caveat that you need to be able to bite off only as much as you can chew. So don't plant, say, oh, well, I want to do a really good job, so I'm going to plant 5 acres of my seed crop for stock seed and walk every row and only end up with and eliminate 95% of the plants in this field, because if you get busy and you run out of time and you're only able to walk half of that field, and the other half you weren't able to do selections in, then you shot yourself in the foot, because you still have all of this pollen coming and all of the seed potentially coming from a whole half of the field that you weren't able to select. So grow as large of a population as you can do a good job of handling. As much as possible, it's really important that you try and grow your stock seed in a place that has nice uniform conditions. Think about a good piece of ground on your farm that you know from past experience is relatively uniform throughout it. So that way you, again, as much as possible, when you're looking at plants, you want to give yourself every opportunity to really be seeing genetic differences instead of differences just due to the fact that one plant was growing in the shade versus the sun, or in a old gravelly creek bed versus in some thick loam. And then finally, another trick that you can do is you can do something called selecting from quadrants. And what I mean by this is, if you look at this picture here, imagine this was your field. Rather than just selecting, say you're going to pick the 20 best plants in your field, rather than just picking the 20 best plants from anywhere, mentally divide your field into 4 pieces and try and pick the 5 best plants from one corner, the 5 best from the other, 5 best from this corner, the 5 best from this corner. The idea here is that if there was, say, a gradient of fertility or water availability in this field, and as it got darker, it was more and more favorable. In this scenario, you're still picking, say, the plants that were the 5 best in the really fertile conditions of your field, but you're also picking the 5 that look the best in the least fertile conditions. And so this is a way that you can be selecting plants based on sort of their relative performance compared to plants that were growing in a similar area and likely in similar conditions. So again, dividing the field into mentally into smaller pieces and then selecting evenly from each of those pieces is another way that you can try and tease out and separate which plants really are genetically superior from which ones are just benefiting from superior field conditions. So the last tool in your toolbox for selection is an idea of family or progeny selection, and this is really being able to take your selection to the next level. This is what you can do if you want to make some fairly rapid improvements in your variety and you're willing to invest some time and labor. So I'm going to walk you through what exactly family or progeny selection looks like. Essentially, it's an opportunity to turn what was you just picking individual plants from your variety and trying to figure out which ones that were the best, to really turning it into something closer to the trials that Lori was talking about. So your first step is still going to just be doing mass selection. So say you walk through the field and you find your 20 best plants in your field, and you save seed from those 20 best plants. But rather than just saving seed and putting them all that seed together in one big bag, instead what you're going to do is you would take seed from each of those 20 bags, those 20 plants, and put them in 20 separate bags. So put seed from one plant in a bag that you would label number 1, and seed from the second plant in a bag that you'd labeled number 2, and so on, all the way up till bag number 20 that came from the 20th plant. So now you have 20 bags of seed. The next year, what you're going to grow, rather than growing just a big field, you're going to grow, in essence, a variety trial where you've got 20 different families, as we would call them, that all came from all the seeds in each of those families came from the same mother plant the previous year. So you'll grow separate rows just like you were growing a trial, and you can replicate this just like you were growing a trial, where one row would be plants that were grown from seed that had all come from a single plant the previous year. And the second row would all be grown from a seed that came from your second plant the previous year, and so on. You've got 20 individual rows, or 40 or 60 individual rows if you're replicating, that now you can look at a whole row and you can see, how does this family look as a whole? You really start to see the genetic characteristics once you can grow multiple plants that are all related to each other like this. And if you can grow them in multiple parts in the field, now you're able to tease out if one of the families looks really good in all the locations that it was grown in a field, then you know that it really has something in its genetics that's making it superior. So what you would do then is walk your field where you have these rows that are each planted from separate families, and first start by so the best family. So these are the families where the vast majority of the plants that were grown all look really good, and then go through and eliminate all the plants in the poor families. So, rototilling these, cutting these, pulling these out, somehow eliminating all the plants in the families where the plants didn't look good. And this includes, even if you've got a single, maybe you've got one really pretty nice looking plant in a family, but all of its sibling plants, all of the other plants that are in that same row that came from the same bag of seed look really bad. Don't save that single plant, because you can see it shares genetics with all of these other plants that don't look as good. So eliminate the entire family if it doesn't look good, and only select families where the family, on average, all the plants look good. And then take the second step and eliminate the inferior plants even from your best families. So now you're only saving the best seed, only saving seed from the best plants in your best families. And this is really an opportunity to jumpstart and fairly rapidly improve your variety. So that's selection, looking at it from mass selection and family selection, and how you might implement that in your seed production. I'd like to turn it over to Alice, who's going to be taking some questions that Lori and I can answer.
Okay, great. Thank you. Also, if you have general questions about organic farming, you're always welcome to use the eExtension Ask an Expert service, and someone will get back to you. So moving on to the questions here. In your opinion, do you think it's more important to have within-farm replication or between-farm replication?
I would say that to some extent that depends on what your goal is for the variety. If this is something that you're trying to select for your own on-farm use, then that's where you'll want to replicate within farm. The generic answer is that it's actually generally better to replicate more across different farms. That being said, that's hard to do. It's typically going to be easier for you to put in a few more plots, another couple of rows where you already have an existing trial, than it is for you to add a whole nother trial site somewhere else. So there's sort of the cost-benefit analysis of, if you can afford to add trials at different locations and do a good job of going out to those locations, or working with another farmer to do a good job of evaluating the trials at those locations, then that's going to give you some really useful information, because they might have a slightly different climate, cultural practices, soil, etc., and you'll just be getting some really useful information there. But that's going to require those, you're going to need to make sure that you have the resources to do a good job of that. And if you can't do that, but if it's relatively easy to just add another few more rows to your trial that you've already got going on, then it's better to do a good job than to try and add another location where you might not actually be able to do a good job.
Okay. Let's see. At what point during a plant growth stage would you make the family selection determination? I know you mentioned before that you try and do it before pollination, but do you have any other advice about that?
Yeah, so I would say that's correct. Do it before pollination. Obviously that's easier said than done. It depends on what the crop is, right? So some crops you can do that relatively easy if it's something that you're really selecting primarily based on its vegetative characteristics, something like spinach or lettuce or carrots. All of these you really can look at what the final product's going to look like before it pollinates. But then something like corn or squash, you're looking at many of the things that you really care about, you're going to be seeing after pollination.
Yeah, this person specifically meant tomatoes, so obviously, yeah, it's different.
Yeah.
I'll jump in here too. You can always unselect. So I recommend it depends on what your goals are and what's important to you, but I would say select at various stages. And I will often do selection at whatever stage. If I'm looking for fruit quality, I'll make selections, or if I'm looking for head quality in lettuce, I'll make selections. I'll even make selections early, before it's fully headed or before it's fully ripe. Like if I see a plant that's really vigorous before it's even set fruit, I might flag that plant. But then I'm going to keep an eye on it, and if later on, let's say its fruit set is really low, then I'm going to take the flag away. And you can either use the flags that are on metal stakes, or you can just use flagging tape. One sort of benefit of flagging tape, you can write notes on flagging tape with a Sharpie and tie it on. You can write a note about what you liked about it. And it's also a good idea to somewhat over-select. If you want to end up with 100 plants, it's not a bad idea to select 120, because the plants, let's say you select them in a vegetative phase, but then, and this wouldn't necessarily apply to tomatoes, but something like spinach or lettuce, you might make your selections, but then it might turn out that it's a weak seed producer, or it gets really tall, but then it flops over and it doesn't have a lot of plant architectural integrity in the seed phase. So you can always, not to say you want to go out and just select everything, but you can always par back your selections. So with tomatoes, if you're looking for fruit quality or fruit set, you can select early for architecture, and disease resistance, and then you can assess quality and do another selection later on.
Okay. Yeah, we've kind of got to get through another bunch of questions in the next 5 minutes or so, so let's try to do that here. Let's see. In how many years do you consider a plant to be a landrace plant, and what about cross-pollination that makes a better plant with better fruit?
I don't have and Lori, feel free to jump in. I don't have a clear definition of how many years something is before it's a landrace. Typically, when we'll talk about landraces, this is something that is developed over a long period, and what that period is, I don't think is clearly defined, but is typically also associated with a particular area. So this may be something like landrace dry beans in the Southwest, that where these varieties may have been selected and adapted to that those environmental conditions for hundreds of years potentially. But I can't say a clear number on how long it is before something gets the title landrace or not. As far as the question of, can you repeat the question about cross-pollination?
Yeah, this person just is asking, what about cross-pollination that makes a better plant with better fruit?
I think maybe what that's referring to is, so in cross-pollinated crops, and we didn't get super thick into the genetics of it. I think Lori may have covered some of this in the last webinar, but with many cross-pollinated crops, they benefit from having a diversity of genes within them. So they benefit from cross-pollination, and on the flip side, they suffer from having too little diversity. They begin to have what we call inbreeding depression. So talking about something like corn, if you just saved seed from a single plant of corn and it didn't have an opportunity to cross-pollinate with anything else, and you did that for several years, what you notice over the course of those years is that the plants that grew from the seed you were saving would get smaller, the ears of corn would get smaller, and that's due to something known as inbreeding depression. That doesn't exist for all plants, but it does exist for many of the cross-pollinating crops. So the flip side of that is something we call heterosis or hybrid vigor, where when plants that benefit from cross-pollination do get cross-pollinated, then they tend to have better fruit size, better seed set, larger plants, and in subsequent generations. And so there's always sort of a balancing act between the level of selection that you apply in a crop that can suffer from inbreeding depression. How many plants do you end up saving seed from? How many do you eliminate, so that you're really selecting from the best while not reducing the genetic diversity so much in your variety that it begins to suffer. And those numbers aren't set in stone. If you look at some of Organic Seed Alliance's publications, like our Basic Seed Saving Guide, it has some decent baseline numbers for how many plants you would want to ultimately save seed from to reduce the risks of losing genetic diversity and creating inbreeding depression in your crops.
Yeah, I just wanted to bring everybody's attention to those resources. If you go to seedalliance.org, the website of the Organic Seed Alliance, you can find a lot more information. Also on the eExtension website. But we have a question about from somebody specifically about where they might find information about tropical seed saving in tropical climates.
I'm trying to think of where some of the best resources are for seed saving in the tropics. So there are, though not strictly the tropics, there are some good publications for seed saving in the southeastern United States that Jeff McCormick wrote, that I think are available through the Carolina Farm Stewardship Association. And so some of those challenges that they have in growing seed in the southeast can apply to other warm, humid locations. There's the Hawaii Public Seed Initiative that you can look up that has some additional resources and knowledge around producing seed in Hawaii, which again, depending on which island and what part of what island you're talking about, can also have some tropical type conditions on it. Those are the two that come to the top of my mind. Lori, I don't know if you have anything else.
Yeah, I don't. That's not one I know too much about. Another idea I had is check out the international germplasm stations. I forget what the tropical one is, like ICARTA.
Okay, well, we can try to get back to that person after the webinar is over with some additional resources, because I think that is a great question. If one family has many traits that you like, but poor seed set, and you don't have as many individuals as your other families, how might you determine if it's still worth working on that family?
That's a great question, because the reality is that you are often not going to find the perfect combination of everything in a single plant or in a single family. So how do you balance that? And so part of that is just going to have to be, first off, sort of mentally thinking about what your priorities are. Just how important is each trait, and how important is it, if there is something that's sort of a do or die trait where you say, I really, this is with the example that question asker had, if seed set is critically important for you, and you say, well, this sure does look nice, but its seed set is really poor, yeah, it's not going to be cut and dry, because you could say, well, how much poorer is it than the other families that you might have, and how much better is it for all the other traits than the other families? Because if you're talking about and how important is seed set, and then finally is, are you working in something that's a self-pollinating crop or a cross-pollinating crop? Because if you are working in a cross-pollinating crop, you might say, well, this does have slightly lower seed set than everything else, but it is just so nice in every other way that I'm willing to allow it to cross back together with the other families, and expecting that in subsequent years I'm going to be seeing something that's a little bit more of an intermediate between the family that has all of these quality traits that make it superior but slightly lower seed set, and the other families and if you feel like, well, I'd be happy with something ultimately that was an intermediate of that could be okay. If it's a self-pollinating crop, then you might essentially, the family that you have, it's sort of what you're going to end up with. It's not, unless you intentionally make crosses, it's not going to cross with the other families out there. And so you have to decide, is this, if I look at this on its own merits, is this something that I would be willing to grow again if it had this poor seed set, even if it looked this pretty? If it's not something you'd be willing to grow or think you could profitably grow, then it may not really be worth keeping.
One thing that my initial instinct was, if it's something that outcrosses, to find something else that's as similar in the suite of qualities you're looking for and cross it to something that has, or allow it to pollinate, cross-pollinate with something that has better seed set. And then another thought I had was whether there's something you can do to increase the pollination and fertilization to increase your seed set, because it may be genetic, but it may also have an environmental component. So can you put it in a hoop house if it needs more heat? Can you buy pollinators if it's something that benefits from insect pollination? Can you put it somewhere where there's more insect activity around, or there's wind, which might increase pollen flow or jostling the flowers? Or are there physical and environmental things that you could change to increase your seed set? Because if it's something you really love and it got just the right suite of characteristics, there may be other techniques to boost your seed production.
Great. We are running out of time, unfortunately, but I would like to thank everybody for their questions and encourage you to attend the other webinars that are coming up in this series on the third Tuesday of each month through November. And we are recording all of them, and they're all going to be archived at the link on your screen as well as on the eOrganic YouTube channel. So if there's one that you can't make, you don't have to worry, you can watch it later. So I'd like to thank you, Jared and Lori, for this presentation, and thanks to everyone for your questions and for joining us.